we have discussed about the introduction and what are the parts involving in alimentary canal and about the digestive glands. Now we will see the real process of digestion. Okay, we all know digestion is of mechanical and chemical process. Now we will start with the buccal cavity. In buccal cavity there are two events taking place, which is mastication and swallowing. Mastication is nothing but making the food, chewing it well, that is the mastication, and we will be proceeding with the swallowing. Now this teeth and tongue which is present in the buccal cavity will be helping us in mastication of the food. What will be happening there? The saliva. Okay, we know in the buccal cavity we are getting saliva from the salivary glands, as we discussed here. Now in the saliva we have something called mucus. This mucus will be helping the masticated food to lubricate and other together, which is nothing but it is called as bolus. What is a lubrication? Lubrication is like making it more liquidy, which is easier to swallow, and other is like joining of all the food particle. So we are doing mastication here, and with the help of saliva we are converting the masticated food particle into bolus. Now this bolus will be going through the pharynx. From pharynx it will be reaching the esophagus. So from the buccal cavity we are doing mastication, and mastication saliva is getting mixed and it is making as a form into the bolus. The bolus will be moving to the pharynx. From pharynx it is started going into the esophagus. This is called deglutition. Mastication and deglutition. The next step we are seeing is peristalsis. After it reaching the esophagus, we will be having the successive waves of muscular contraction happening in the esophagus, which will be helping the bolus to move throughout the esophagus. That is called as peristalsis. So from esophagus we will be reaching to the stomach. The food will be reaching to the stomach, and it will be guarded by the gastroesophageal sphincter, which we already discussed in the previous video. So this is the initiation of food, where mastication, deglutition and peristalsis happen. So coming to the next one. In the saliva, we already talked about the saliva, right? So what are the things we are having in the saliva? In saliva, we have electrolytes, enzymes, salivary amylase and lysosomes. The chemical process, we talked about the mechanical and chemical method in the digestion. Here the chemical process is firstly beginning with the salivary amylase. That's what I mentioned here. So if you see, the first chemical process is happening in the oral cavity, which is nothing but the buccal cavity. What is happening there? The salivary amylase, which is the carbolytic splitting enzyme, by its hydrochloric acid, it will be converting the disaccharides to the monosaccharides, which is nothing but, if you see, this is the example for the starch, and at the pH 6.8 using the salivary amylase, it will be converting the starch to maltose. This is the first chemical process. What is the chemical process? Reaction happening by an enzyme. Here the enzyme is the salivary amylase, and only 30% of starch is getting converted into maltase here. This is the important point, not a whole starch. And then we have lysosome, which is the antibacterial agent. That's why if we get a wound, some people suggest us to keep the saliva over there, because the saliva is having the antibacterial property. Now here we stopped till the stomach. As we all know, there are 4 layers in the alimentary canal, and mucosa is the innermost layer. In mucosa, we also discussed that we have gastric glands in the stomach. So if you see, so yeah, coming back to the point, what are the components we are having here, or cells? We have mucous neck cell, which secretes the mucus, and peptic or chief cells, which is the proenzyme pepsinogen. Proenzyme means it is in active form. Then inactive form. Then partial oxygenating cell, which provides HCl and intrinsic factor. What is this factor? This factor is an essential factor which will absorb the vitamin B12. This is the use of intrinsic factor. Now we talked about what are the cells present in the mucosal layer of the stomach. Now coming to the next one. Now the bolus from the esophagus will be saved into the stomach for 4 to 5 hours. What will happen in the stomach? In the 4 to 5 hours, it will be mixes the bolus, the food we get, with the acetic acid juice. How it will be mixed? By the chyme. Here we talked about bolus, which is the masticated and lubricated food. Here it is chyme. Chyme is nothing but the churning movements of muscular wall. It will be keep on moving the food with the churning movement, the food will be getting indulged into the acetic gastric juice, which we talked here as the HCl. Okay. Now, when this HCl is acted upon the proenzyme pepsinogen, which is secreted by peptic or chief cells, it will be converted to the pepsin, which is the active form. As I said earlier, proenzyme is the inactive form. When it is getting mixed with the acetic gastric juice HCl, it will be converting into the active form pepsin, and this is the proteolytic enzyme. What it means when the proteolytic enzyme means, it will be splitting the proteins. That is the meaning. Here the pH will be 1.8 for the HCl. So, and we also talked about the mucus present in the stomach. Mucus along with the bicarbonate, what will it will do? It will lubricate the food. That will make us easy for the digestion and also protect the mucosal epithelium from HCl. That's why when we are in the empty stomach, we will be having the burning feel due to the HCl. That's why this mucus will be protecting the mucosal epithelium from getting affected from the HCl. So then we have another protein called renin, which is also similar to the pepsin. Pepsin will be act on proteins. The renin, which is present in the infants, will be act on the milk protein that is present in the infants. And we have some small amounts of lipases also secreted in the stomach. So we saw from the buccal cavity, mastication and lubrications is happening. It is getting converted into bolus. From the bolus is further moving into pharynx, esophagus. From pharynx we are reaching the esophagus we are reaching the stomach, and in stomach what are the cells present and how it is getting acted. Now we are done with the stomach. So from stomach next it will be moving to the small intestine. So what are the juices present in the small intestine? We have three: bile juice, pancreatic juice and intestinal juice. All these three will be getting into the small intestine. In here, bile juice and pancreatic juice, they both will be entering into the small intestine through a common portal, which is the hepatopancreatic duct, and we have intestinal juice along with them. Now let's see what are the components in the pancreatic juice. The pancreatic juice contains inactive enzymes such as trypsinogen, chymotrypsinogen, procarboxypeptidase and amylases, lipases and nucleases. All these are inactive enzymes present in the pancreatic juice. Now the trypsinogen in the pancreatic juice will be acted by the enterokinase, which will be turning the inactive trypsinogen to trypsin, which is an active form. From where we are getting this enterokinase? It will be present in the intestinal mucosa. Trypsinogen is converted into trypsin by enterokinase, and it is present in the intestinal mucosa. This trypsinogen in turn active other enzymes which are inactive in the pancreatic juice. Clear still now? Let's start about the bile juice. Bile juice have bile pigments, which is bilirubin and biliverdin. Apart from this, we have bile salts, cholesterol and phospholipids also, and there is no enzyme present in the bile. So we are done with pancreatic juice, bile juice. Let's move on to the next one. This bile will help us in emulsification of fats. That is, this bile will be helping us to breaking down the fats into small micelles. We talked about carbohydrates, protein and fats, which are the biomacromolecules, and we have the food in the major content. So here that bile will be helping us the fat to convert into the small micelles, and it is also helping us in the activating the lipases. We are now going to see the intestine. Okay, in intestine, the mucosal layer contains the goblet cells, which we already discussed here. When we are talking about the alimentary canal, layers of alimentary canal, we talked about the goblet cells. The same thing I am stating here. So yeah, in the intestine of the mucosa, we have goblet cells which secrete the mucus, which help in the lubricating, as you all know. And also we have brush border cells of mucosa plus goblet cells. We have also discussed about the brush border cells in the villi. Okay, so what they are saying, this brush border cells of mucosa but goblet cells will form the intestinal juice with also the circus entericus. What is circus entericus? It consists of brush border cells of mucosa and goblet cells. Together it is the intestinal juice. Now, what are the components present here? We have disaccharidases, dipeptidases, lipases and nucleosidases. Next. So here also we are mucus and bicarbonates protecting the intestinal mucosa. If you remember, in stomach also we have seen the same point. that the mucus and bicarbonate will be protecting the mucosal epithelium from HCl. Here also in the intestine the same thing is happening, and apart from the acid it is also protecting from the alkaline, which is of pH 7.8. And we are Werner's gland is also present in the submucosal layer of the intestine. That is, in the submucosal layer of the small intestine we have Werner's gland, which is also helping on the same. Still clear with these things? Like we talked about the pancreatic juice, intestinal juice and bile juice. Now we'll see what are the conversions happening here. They're really important. Proteins, peptones and proteases, which are from the stomach, are getting converted into dipeptides. These are the first step. These are not the final form. These are happening in the first stage. Okay. Dipeptide. How it is all getting converted? It is all getting converted from the pancreatic enzymes such as trypsin, chymotrypsin and carboxypeptidases. Now coming to the next one, the starch. Only 30% of the starch is getting into maltose in the buccal cavity. So the remaining starch here will be acted by amylase, which is also coming from the pancreatic juice, and it will be converted as disaccharide. Then coming to the fats, it will be first converted into diglycerol, from then monoglycerol. All these are happening by the bile juice, which is the giving the enzyme lipase to ensure this process. Now the nucleic acid present here will be converted into nucleotides and nucleosides with the help of nucleases, which is also an enzyme from the pancreatic juice. So these are the major conversion which is happening from the stomach with the help of pancreatic and bile juice. And if you see, intestinal juice acts on the final products. These are the final product that we receive now. Now the intestinal juice, we see here, this intestinal juice will act on all the final product, and we will be getting the simplest form which will be absorbed by our body. And these things will be happening closest to the mucosal epithelial cells in the intestine. So in this video we talked about how the digestion is starting, from where it is starting. We talked about the buccal cavity, saliva, mastication, deglutition and the lubrication, and it is converting up into bolus. That bolus is going to the esophagus. From esophagus it will be reaching the stomach, and we talked about what are the cells there in the stomach and what are the uses of it and what are the actions taking part in it. It is first converted into chyme. The chyme is acted upon by the enzymes present in the cells, and coming from there we talked about small intestine, what are the juices coming from here, and we explained about each of the pancreatic juice and bile juice and what are the uses, and we talked about the conversion. And in next video we will be talking about the intestinal juice and how it is acting on all this product.